In this mini video, I will be discussing epistasis and how multiple different genotypes can lead to the same phenotype. First off, what is epistasis? Epistasis is when the expression of alleles at one locus masks or modifies the expression of alleles at a different loci. In other words, genes at different loci interact unexpectedly to determine a single phenotype. One term to recognize is that the gene that does the masking is called the epistatic gene. To help further explain epistasis, we're going to use man's best friend as an example. If you couldn't tell by the pictures of my dogs, we'll be looking at how epistasis plays a role in a dog's coat color. To begin, we need to understand the basics. First off, we will be looking at 3 different coat colors, brown, black, and white. In this example, we will also be looking at 2 different alleles, A and B, which will each contribute to the genotype and thus the phenotype of each dog. Dominant alleles will be capitalized and recessive will be lowercase. Looking at the specific genotypes of each dog, brown dogs must be homozygous dominant or heterozygous for the A allele to exhibit a brown coat color. Any dog with a dominant A allele must be brown. Now looking at a black dog, it must be homozygous recessive for the A allele and have at least one dominant B allele to have a black coat color. For a dog to have a white coat color, it must be homozygous recessive for both the A and B alleles. Now let's look at the specific kind of epistasis we're working with, as there are 6 different modes of epistasis. For our example, we will be looking at single dominant epistasis. In this mode of epistasis, a dominant allele at one locus will mask or modify the expression of alleles at a second locus. So in our example, the A allele is our epistatic gene and will always be expressed over the B allele if the dominant A allele is present. With this information in mind, let's look at the epistasis pathway of coat colors in dogs. Starting from the left, if an animal has at least one dominant A allele, they will automatically express the brown coat color and thus not progress further down the pathway. If the dog doesn't have a dominant A allele, meaning it is homozygous recessive for the A allele, it has the possibility of having a black or white coat coloring. Looking at the pathway, if the dog is both homozygous recessive for both the A and B allele, it will have a white coat color. If it does possess a dominant B allele and is homozygous recessive for the A allele, it will have a black coat color. Let's put this pathway to the test with some dihybrid crosses. First, we're going to make 2 brown dogs that are heterozygous for both the A and B allele. After performing the cross, we had 12 dogs with a brown coat color, 3 dogs with a black coat color, and 1 dog with a white coat color. As you may have seen in the epistasis ratio chart previously, this 12:3:1 ratio aligns perfectly with that of single dominant epistasis. Also, take note that each phenotype has different genotypes. Next, we're going to make a brown dog with a black dog. Make sure to pay special attention to the genotype of each dog before performing the cross. After crossing, half the progeny should be brown and half should be black to create the ratio 8:8 or 1:1. Again, this ratio confirms that our mode of epistasis is single dominant because the presence of a dominant A allele in half our progeny results in half our dogs with a brown coat color. The other half of the progeny is homozygous recessive for the A allele and contains at least one dominant B allele, leaving the other half of our progeny with a black coat color. Thank you for listening, and I hope you had a barking good time learning about epistasis with my furry friends.